Students, next concept in applications of biotechnology, that is development of BT cotton. Here BT means Bacillus thuringiensis. Bacillus thuringiensis is the bacteria. Bacillus thuringiensis is the bacterium. From that bacteria, gene is extracted. Genes which are extracted. Such genes which are incorporated into the cotton plant. The genes which are incorporated into the cotton plant. Then cotton plant genome is altered. The cotton plant genome is altered. After that, the cotton plant is said to be genetically modified cotton plant, or genetically engineered plant is produced by the insertion of foreign gene into the cotton plant. Cotton plant becomes genetically modified. Its genome is altered due to the insertion of foreign gene. Like that, the plants which are obtained, like that, the organisms which are obtained, that organisms are called genetically modified organisms. Through the process of biotechnology, one of the plant is developed. That plant is highly resistant to insects. Highly resistant to insect. The insect resistant cotton plant is developed. That cotton plant is called as Bacillus thuringiensis cotton. Bacillus thuringiensis. Here, one of the bacteria is used for the production of cotton plant. That bacteria is called as Bacillus thuringiensis. Bacillus thuringiensis is the bacteria. That Bacillus thuringiensis usually produces a protein. Bacillus thuringiensis produces a protein. That protein kills some of the insects. The protein produced from the Bacillus thuringiensis usually kills some of the insects. Like lepidopterians, coleopterians, and diphterians. Here, lepidopterians includes budworms, and armyworms. Coleopterians includes beetles. Diphterians includes flies and mosquitoes. Usually, Bacillus thuringiensis is the bacteria. That bacteria produces a protein in its metabolism. That protein usually kills some of the insects like lepidopterians, coleopterians, and diphterians. And Bacillus thuringiensis produces protein crystals. That protein crystals which contain toxic insecticidal proteins. Bacillus thuringiensis bacteria produces a protein crystals which contain toxic insecticidal protein. The Bacillus thuringiensis usually produces a BT toxin. BT toxin. Bt toxin. Initially, the Bt toxin initially is inactive. Inactive protein. Inactive protein. The inactive toxin, inactive protein or inactive toxin, that is called as protoxin. Protoxin. Bacillus thuringiensis produces a protein toxin or Bt toxin. That Bt toxin initially inactive toxin. That inactive toxin called protoxin. That protoxin ingested by insects. The protoxin is entered into the gut portion of the insect. The protoxin, inactive toxin, is ingested by insect. That the protoxin is entered into the gut region. At the region of the gut portion, where there is an occurrence of alkaline pH. In presence of that alkaline pH, the protoxin is dissolved. Protoxin is dissolved, particularly at the region of gut portion in presence of alkaline pH. Protoxin is solubilized. At the region of the gut in presence of alkaline pH, and afterwards protoxin becomes active toxin. That active toxin affects the epithelial tissue present at the region of gut region. Usually, that active toxin damages the epithelial cells, and that causes swelling of epithelial cells, lyses the epithelial cells. Then there is a formation of pores or holes at the region of gut portion, and afterwards insect becomes death. Death of insect takes place due to the conversion of protoxin into active toxin in presence of alkaline pH, particularly at the region of gut portion. The epithelial cells, epithelial tissue present at the region of gut portion, epithelial cells which are totally damaged, then the insect becomes dead. Now, due to this condition, what is that? Is all the insects which are destroyed by the activity of active toxin. Here, that protoxin, which are produced by protoxin, is encoded by genes. Protoxin encoding genes are called as cry genes. The cry genes which are extracted from the bacteria, that is Bacillus thuringiensis bacteria. The cry genes which are extracted from protein, it means Bt toxin producing genes present in a Bacillus thuringiensis bacteria. Such genes which are considered as cry genes, that cry genes which are extracted from Bacillus, isolated from Bacillus thuringiensis, such genes which are incorporated into crop plants like cotton plant. And after insertion of gene into the cotton plant, that cotton plant becomes genetically modified or genetically engineered cotton plant is developed due to the insertion of Bt toxin encoding genes present in the bacteria that should be extracted from the bacterial cell. Such genes which are inserted into the cotton plant, then the plant is genetically modified. Such plants which are raised in the process of biotechnological process. Here, the protein encoding genes, Pt toxin encoding genes are called as cry-genes. Cry-genes. Here, the cry-genes which are occur in various types. Cry-genes which are occur in the form of various types. Here, cry1AC and cry2AB. Cry1AC and cry2AB usually kills bollworms. Usually this kind of genes which produces a beta-toxin which kills bollworms. And cry2AB usually destroys carnivores. Like that. Here the genes which are extracted from the bacteria, such genes which are inserted into the cotton plant. That plant which always resist to some of the insects like lepidopterans, coleopterans and diphterians. Through the process of biotechnological process, the plant is raised. That plant is called as BT. Cotton plant is raised. Like that of BT cotton development, through the process of biotechnology, pest resistant plants can be raised. Pest resistant crop plants can be raised through the process of biotechnology. One of the nematode called malloid gain incarnation is the nematode. Usually that malloid gain incarnation infect root system of tobacco plant. Due to that infection, tobacco plant reduces the productivity, productivity. To avoid, the infection can be prevented. By using RNAi. Usually nematode malloid incarnation infect the root system of the tobacco plant, hence productivity is decreases, reduced. To avoid this infection, to prevent the infection, by using RNAi. Here RNAi means RNA interference. RNA interference is used to avoid the infection of malloid gain incarnation. Usually that RNA interference silencing mRNA to form dsRNA, double-stranded RNA. RNA interference silencing the mRNA to form dsRNA, double-stranded RNA. That should be prevent translation of mRNA. That the major function of RNAi. Usually, that nematode specific RNA, nematode specific RNAi gene, nematode specific RNAi producing gene extracted from the host cell. That RNAi producing gene incorporated into the plants like tobacco plant by using agrobacterium vector. Agrobacterium acts as a vector. The transfer of nematode specific RNA producing gene into the tobacco plant by using agrobacterium vector. Then after the insertion of gene RNA producing gene into the tobacco plant, it becomes genetically modified plant. This RNAi gene, usually in this plant, tobacco plant, RNAi produces sense and antisense strands and silencing the mRNA to form dsRNA. When the dsRNA double-stranded RNA is formed, ultimately translation process stops in a host cells. So that RNAi silencing the mRNA in a nematode. RNAi silencing the mRNA of the nematode. Hence, the nematode, it means myeloid vein incarnatia, will not survive in a tobacco plant. RNAi producing genes which are extracted from the host cell, which are incorporated into that tobacco plant by using agrobacterium vector. That RNAi genes which are inserted into the tobacco plant, which produces sense strands and antisense strand, which causes the formation of double-stranded RNA. And in a tobacco plant, nematode will not survive. With the help of the process of biotechnology process, what is that? Myeloid incarnatia can be stopped by insertion of RNAi gene. RNAi means RNA interference. RNA interference usually silencing the mRNA